5H, blood typing. As you recall from our lesson on the cell and the cell membrane, cells will often have surface proteins or glycoproteins, which are used for communication and identification by the cell. Some of these markers are important as they label your cells belonging to yourself. This allows for your body to recognize foreign cells that may find their way into your body and to destroy them, while leaving yours alone. This defensive feature to ensure that harmful things like bacteria, viruses, toxins, and tumor cells can't enter your body freely. Any markers that result in an immune response is called an antigen. Special leukocytes called B cells help to identify foreign cells and will produce particles in response to the antigens called antibodies. These antibodies are important as they cover the antigens of the pathogen, preventing the pathogen from using them like keys to enter host cells and infecting them. The antibodies also label the pathogen to be destroyed, often by macrophages, the eating leukocyte. Another benefit of the antibodies are the binding of multiple pathogens together by their antigens, which keeps them from being able to move around and makes it easier for the macrophages to digest the pathogen. As you are probably aware, we have to be very careful of the blood that we receive from donors, and that is because of the antigens that our red blood cells have on their surfaces. These antigens act as license plates, indicating that the red blood cell belongs to you. There are only two antigens, or license plates, that we find on red blood cells. They are the A antigen and the B antigen. These are determined by your genetics, and you may have been born with red blood cells with the A antigen, B antigen, both the antigens, or neither of the antigens, which we call blood type O. If you have blood type A, you have antigen A, similar to a license plate labeled A. This means that persons with blood type A would carry antibodies B, protecting them from any blood type B from entering their bodies. Therefore, they can only receive blood type A, their own, or O, which has no surface antigens at all, or no license plates. Blood type B will have B antigens and A antibodies. Blood type B will have B antigens and A antibodies circulating through their system. Blood type AB will have both antigens A and B, so their immune system will not have any antibodies for either A or B. They would simply end up attacking their own blood if they did. This means they can accept any blood type, which makes them the universal acceptor. Blood type O has no antigens. Therefore, this person would contain both antibodies A and B, which means their immune system would respond to any blood that has either surface protein on it. They can receive only blood type O. However, due to the lack of surface markers on their blood, they may donate to all blood types, making O the universal donor. What would occur if someone was given the wrong blood type? Well, a process called agglutination would occur. This is where a person's antibodies would bind the foreign antigens, resulting in clumping of red blood cells, which would essentially lead to clots. This would lead to major organ issues. Survival from agglutination from receiving the incorrect blood type is very slim. Here you can see red blood cells agglutinate. Here you can see the clumping or agglutination of red blood cells. Blood that has agglutinated takes on a grainy appearance. Lastly, there is a second surface marker called the rhesus factor. This protein is not from the same gene as the A and B antigens, but they do produce an immune response if the rhesus negative person receives the positive blood. If a person's blood includes this protein, they are said to be positive. Hence the term A positive versus A negative. If the rhesus factor is included in the count of different blood types, we have a total of 8 possible combinations.